Okay, so for the first expression, for the first question, what we're gonna do is overexpression of BRCA1. To do that, we're gonna transfect a virus promoter plasmid. So we're gonna use a virus promoter. First with our control gene. And then for the second one, we're gonna do virus promoter with BRCA1. So after we do that, we're gonna have 293 T cells, I think. So we're gonna have two of those. We're gonna transfect our control, and then our BRCA1 overexpression plasmid. After we transfect that, we're gonna wait 24 hours. After we wait 24 hours, what we're gonna do is to do Western blot, first we have to isolate the protein. We isolate the protein and run gel. So in our gel, this is gonna be our ladder. And then here it's gonna be our control and BRCA1. And then after that, after we run our gel, we're gonna transfer it into a membrane. And then after we transfer it to a membrane, we're gonna assume this is the membrane instead of the gel right now. This is the ladder. We're gonna add antibodies. So what antibody are we adding? We're adding, well, let's just say we can do for the primary antibody, we can do mouse anti-BRCA1. And then we're gonna add a lot more in the BRCA1 compared to the control one, because we expect a lot of BRCA1 in the BRCA overexpression. And then after we do that, we're gonna add our secondary antibody. We can call that horse anti-mouse. So this will help us see it better. And then after we do that, we're gonna run a Western blot. And then in our Western blot, this is our Western blot. For the control, we should expect BRCA protein. But for our BRCA1 overexpression, where we added overexpression, we should expect a lot more BRCA1. So that'll be for the first question. The second question is asking, we now have cells that have no BRCA1. Basically, how we can achieve that in the first place is, say we have cells here. We can do a control if we want. Control siRNA and BRCA1 siRNA. So we can transfect the siRNA against BRCA1, and then we'll just leave this here. We can just do the BRCA siRNA, because since most of the questions are there. So after we do that, we're gonna wait 24 hours, and then we're gonna treat the cell with Taxol, and then wait another 24 hours. After this, to see the flow cytometry, basically, what we're gonna do is we're gonna permealize the cell first, and then after we do that, we're gonna stain the DNA. How we use propydium iodide? Yeah, so we use propydium iodide. We stain the DNA. After that, we can do the flow cytometry. Before that, to check if to check the BRCA siRNA, we can always do Western blot. And we should expect small amount of BRCA compared to the control. But the flow cytometry should be the same. Should be the same to the normal flow cytometry. Why is that? I'm gonna put amount of cell, amount of DNA. Actually, number of cell. Why is that we expect the normal flow cytometry? Because reduction of BRCA siRNA, or if there's reduction of BRCA1, or meaning that there's no BRCA1 in the body, or there's a small amount, means that Taxol cannot work properly. The cell basically has a resistance against Taxol. And what does Taxol do? Taxol, what it does is it stops the cell in metaphase. It kind of basically inhibits it from going to anaphase and basically like separating into two daughter cells. But if there's no BRCA siRNA, Taxol won't work. The cell will go on as if nothing happened. So that's why we should expect a normal flow cytometry. That's it. Thank you.